Welcome back to the channel. In this video, we're going to be looking at lipids. We'll start off by talking about how fats differ from oils. We'll go on to the functions and uses of lipids in living organisms. We will talk about triglycerides and the bonds found between glycerol and fatty acids in triglycerides. We will talk about why lipids are not described as polymers. We'll distinguish between saturated and unsaturated fatty acids. We'll talk about what phospholipids are and how they differ from triglycerides. We'll also talk about the role of phospholipids in cell membranes, and then we'll finish off by considering whether or not triglycerides can be used as sources of metabolic water, for instance in desert animals like camels. So let's get on with it. So as usual, in order for you to get the most out of this experience, you should really be filling in the worksheet as we go along. There's a link to the worksheet in the description below. So let's start off with page 1. So there are two main types of lipid, as I've already alluded to. You've got fats and you've got oils. So fats are lipids which are solid at room temperature. And they usually come from animal sources. For example, lard. Okay. And then oils are lipids which are liquid at room temperature. And they usually come from from plant sources. So plant sources, for example, olive oil. Okay, so that's a quick introduction to the difference between fats and oils. So why would we even talk about lipids at all? Basically because they perform lots of different vital functions in living organisms. So let's put down some of those functions. So they're the main components of cell membranes. They can act as buoyancy aids. E.g. in seabirds. They can be used as energy sources, so used as energy storage molecules. They can also be used for things like waterproofing, so for instance on feathers of seabirds like ducks. Okay, so used for waterproofing. E.g. on feathers of ducks. Okay, so let's see if you can come up with another use of lipids in living organisms. And there's a drawing or a diagram on that page which might give you a clue as to another example of a function or use of lipids in living organisms. All right, so on to the next bit. We're going to look at a group of lipids, the triglycerides. So triglycerides are the ones that you've probably come across a GCSE, and they're made up of glycerol and 3 fatty acid molecules. Okay, so you can see there in the diagram that I've provided the structure of a triglyceride. And in that picture, you can see glycerol in pink there. So there's a glycerol molecule. And then you can see the 3 fatty acid molecules. And when they bond to each other, they form a triglyceride on the right-hand side of the arrow. And in order to make the bonds between the glycerol molecules and the fatty acid molecules, you lose water molecules. And if you remember from our look at carbohydrates, that would be an example of a condensation reaction because water is given off when the bond is made. And it says label one of the ester bonds formed by the loss of water molecules on the diagram. So you might want to pause now and see if you can do that. And let's have a look. If you labeled here, then you would be right. So that is an example of a bond within, or a bond which is formed between glycerol and the fatty acids. And those are called ester bonds. Okay. So let's write that in. The name of the type of bond, ester bonds. Okay, and why are lipids described as being hydrophobic? That's because they are insoluble in water. They're water-hating molecules. So insoluble in water. And you can think of them as water-hating. Okay, why aren't lipids described as being polymers? So in our video on polysaccharides, we talked about polysaccharides as polymers. And the reason why they are polymers is because they're made up of multiple monomer molecules. And all the monomers are of the same type. So in a polysaccharide, all the monomers would be monosaccharides. Okay, so why are lipids described as being polymers? And the reason why is simple. They are not made up of the same type of constituent molecules. And in essence, or i.e., or that is, in this case, you end up with glycerol not being the same as fatty acids. Glycerol is not the same as fatty acids. As such, there are 2 different types of molecules which form triglycerides, as an example of lipids, and as such, they can't be described as polysaccharides. Okay, or sorry, as polymers. So let's go on to the next page. So we've got saturated and unsaturated fatty acids. So have a quick look at that diagram and see if you can identify what the differences are between, or what the main difference is between a saturated and an unsaturated fatty acid. So A on the left being a saturated fatty acid, and B on the right being an unsaturated fatty acid. So have a quick look and see if you can figure out what the difference is between those molecules. You can pause now and think about it. Okay, so you probably noticed that in unsaturated molecules, in unsaturated fatty acids, there are one or more double bonds between carbon atoms. And you would not have any double bonds in a saturated fatty acid. Okay, so how do they differ in terms of what happens to them at room temperature and their usual sources? So saturated fatty acids would tend to be solid at room temperature. So saturated fatty acids tend to be solid at room temperature. And sources, yet again, would tend to be animal fats. And examples would be things like lard and butter. Unsaturated fatty acids, on the other hand, tend to be liquid at room temperature. Okay, and sources would tend to be plant lipids. And an example would be something like olive oil. Okay. So let's go on to the next bit. What are phospholipids? These are triglycerides with one fatty acid replaced by a phosphate group. Okay, so you can see there a diagram of a cross-section through a plasma membrane in a cell. And if you look closely, you'll notice that most of the molecules present are phospholipids. They're the yellow squiggly lines with red dots at the top. So what features or components of phospholipids enable them to form bilayers within cell membranes? Well, you've got the hydrophobic tails. Actually, let's get those inverted commas and put them here instead. And the hydrophilic heads. And how are cell membranes in organisms in colder climates kept fluid? The presence of cholesterol. This prevents them solidifying. Okay, so on to the next page. So triglyceride structure. How are the intramolecular bonds in lipids broken down? And they are broken down, as I've mentioned earlier, using a process called hydrolysis. And how does that work? It adds water molecules across bonds to break them. Okay, so what are the main products of the complete breakdown of triglycerides? The main products would be carbon dioxide, water, and energy. Okay, so this seems interesting because if you can get water from breaking down lipids, then maybe that could be an advantage to, say, for instance, an animal in the desert. So, for instance, at the bottom of that page, you've got a camel or a bunch of camels trekking across the desert. So obviously, if water can be derived as a metabolic byproduct of breaking down triglycerides completely, then maybe by having fat in their humps, that might be an advantage because it could supply water as well as energy when they break down lipids. So in theory, this could provide extra water. However, as you've probably deduced by now, I said in theory. In reality, what happens is, yes, molecules of triglycerides produce quite a large amount of water molecules when they're broken down. But the problem is that they also require a lot of oxygen, which means that the organisms would have to take in a lot more oxygen and give out a lot more carbon dioxide, which would therefore require them to breathe faster. And that increased breathing rate would mean that they'd end up losing more water molecules anyway. And as such, overall, there isn't really an advantage. In fact, breaking down some other molecules ends up with a smaller deficit of water molecules being lost. Okay, so unfortunately, the result of breaking down lipids is an increase in net water loss due to gaseous exchange, in essence, and as a result there would actually be eventually a disadvantage at doing this, because you'd end up with even more water being lost than usual, or than from something, for instance, like a carbohydrate. Okay, so that's it on the main content. So let's have a quick summary of what we covered in this video. So we started off by looking at the difference between fats and oils. So fats tend to be solid at room temperature and oils tend to be liquid at room temperature. Fats also tend to be from animal sources, and oils tend to be from plant sources. Lipids have various uses in organisms, which I mentioned as well. So they're the main component of cell membranes. They can be used for things like stabilizing membranes, or well, cholesterol can be used for stabilizing membranes. Lipids can also be used for buoyancy in in animals that need to be less dense in water. So, for instance, seabirds, so that they float. You can also use lipids as an energy source, and so on. We then talked about the structure of triglycerides as a main example of lipids, and triglycerides are made up of glycerol and 3 fatty acid molecules, and they're held together by ester bonds. So how is it that ester bonds are formed? By condensation reactions. And how are they broken down? By hydrolysis, just like when we looked at carbohydrates. We also talked about the fact that lipids are not considered polymers, and that's because, unlike in carbohydrates, so in things like polysaccharides, lipids are not made up of the same or repeating monomers, and as a result they can't be considered as polymers. We then went on to talk about the difference between saturated and unsaturated fatty acids. So saturated fatty acids have no double bonds between the carbon atoms, whereas unsaturated fatty acids would tend to have one or more double bonds between their carbon atoms. The net result of that is that saturated fatty acids tend to result in lipids that are solid at room temperature, and unsaturated fatty acids tend to be liquid at room temperature. We then talked about phospholipids being similar in structure to triglycerides, except basically one of the fatty acids is replaced with a phosphate group. It's phospholipids which form the main component of cell membranes. And it is due to their structure having hydrophobic tails and hydrophilic heads, which cause them to line up in things called bilayers, and that's why they form the main components of membranes. And then finally we talked about the possibility of lipids being used as sources of metabolic water in animals that live in desert areas or places where water is scarce. In those situations, in reality, lipids wouldn't work as a source of metabolic water, because there are disadvantages to using lipids in that way. You end up having to breathe more rapidly, and those increased breaths result in more water loss. So overall, there isn't really an advantage to using lipids, at least not from the point of view of trying to get more water from them. And so that's it really from the point of view of looking at the structure of lipids. As usual, I hope you found that helpful, and if you did, it would be great if you clicked on the like button below, and if you would like to be notified whenever I add more videos, you might want to click on the subscribe button. Good luck, and I hope that you come back to watch future videos.